In this video, we're going to be talking about the digestion of fats. So in our diet, we consume triglycerides, and these are the fats that we consume. So we've got the triglyceride structure here. So the structure is a glycerol, and we've got fatty acids which hang from this glycerol. And I've represented that here with the letters Fa. So now we know what a triglyceride is. We can look at how it gets broken down further in the intestines, which is where this process is happening. So we've got the triglycerides, which are then converted to bile salts and also lipase. And then they become fatty acids plus monoglycerides, which are technically triglycerides because we've just broken down what the original form was into fatty acids plus monoglycerides. And these fatty acids plus monoglycerides together are encapsulated in something called a micelle. So I've got this little green circle here to represent a micelle. So triglycerides, which are within this micelle, so I'm going to angle it here above the triglycerides. Triglycerides come together with cholesterol to form chylomicrons. And once this occurs, these chylomicrons can travel through ducts in the body, so into the bloodstream. So these chylomicrons are a type of lipoprotein. And when they travel through these ducts, where do they end up? They end up in adipose tissue, where fat is stored, and in skeletal muscle, where fat is oxidized or broken down. The enzyme that breaks down the chylomicrons back into the original form of triglycerides is lipoprotein lipase. And we're going to have a look at what inhibits and promotes the secretion of this enzyme in both adipose tissue and skeletal muscle. So let's move this over here. In adipose tissue, we have insulin, which stimulates the expression of LPL in adipose tissue. However, in skeletal muscle, insulin inhibits the secretion of LPL in skeletal muscle. Furthermore, glucagon and epinephrine activate LPL in skeletal muscle. So these are key regulatory points which you should keep in mind. Furthermore, we've got a question here that I'm going to pose. So why is fat stored in adipose tissue but broken down in skeletal muscle? And the answer to this goes back to a topic to do with enzymes. So lipoprotein lipase is an enzyme, and the Km of LPL in skeletal muscle is lower than in adipose tissue. So if we recall that low Km means a high affinity, that means that skeletal muscle is better able to attach on to the chylomicrons and break them down into triglycerides because its Km is lower, meaning it has a high affinity being the ability to grab onto those chylomicrons and break it down. So that's why it's more likely to be oxidized in skeletal muscle, the fat, as opposed to adipose tissue where it is stored. So now we know where our fats are ending up. Let's take a look at what happens to the triglycerides once they get where they need to be. So we have our triglycerides, which are in our chylomicrons, and they can be broken down into the fatty acids and glycerol, because remember, glycerol and fatty acids is what are the components of triglycerides. So we've got our triglycerides, which can become a glycerol or free fatty acids. So they can become the free fatty acids by the enzyme LPL. And LPL is also responsible for making the triglycerides become glycerol. So it's breaking down the triglycerides, so it's separating the glycerol from the free fatty acids. So the free fatty acids hang around in the body, but the glycerol can be broken down further. Just got that left over there. Accidentally picked it up. So let's get started in the first reaction of glycerol breakdown. We've got glycerol becoming glycerol 3-phosphate by glycerol kinase. And in this process, an ATP becomes an ADP. Then we have glycerol 3-phosphate becoming DHAP, which is dihydroxyacetone phosphate. And this is done by glycerol 3-phosphate dehydrogenase. And if you've seen the video on glycolysis, you should know that dihydroxyacetone phosphate, DHAP, a bit of a mouthful, is part of glycolysis and also part of gluconeogenesis. So glycerol can enter glycolysis or gluconeogenesis through this breakdown process. And that's what you need to know about the digestion of fats and how the fats eventually become incorporated into metabolic pathways.